Good morning, everybody, and welcome to the 5th edition of Uncle Ron's Academy. Today we are going to be discussing two very important biological processes known as diffusion and osmosis. Both of these are important because that is how things get into and out of a cell. Good things go in and bad things come out. So to demonstrate first diffusion. Diffusion is a method whereby molecules move from an area of low concentration to an area of high concentration. And they will keep on moving until the concentration on both sides of the membrane are exactly the same. This is also known as dynamic equilibrium. It doesn't mean that diffusion has stopped. It means that the diffusion into and out of the cell is exactly equal. To demonstrate that, we're going to use two small beakers. Here is a beaker of hot water, and here is a beaker of cold water. So the cold water has a temperature of 20 degrees Celsius, which is about room temperature. And we are going to put in a drop of red food coloring. So if you can zoom in on the food coloring and watch what happens to the food coloring as time goes by. We're going to do exactly the same thing to the beaker of hot water. This hot water is approximately 90 degrees Celsius, close to boiling. And we're going to do exactly the same thing. Put in 2 drops. And we're going to see how diffusion works differently when something is cold compared to when something is hot. So if you will notice, in the beaker of hot water, the red food coloring has spread out in a matter of seconds. Whereas in the cold beaker of water, the food coloring has spread out, but there are certain areas that are not finished getting the dye. It's not quite as red as the hot coloring is, proving that diffusion is temperature-based. Something that is warm will make molecules diffuse a lot faster than something that is cold. Molecules get into and out of a cell by diffusion. There is a second process which is related to diffusion, and that is known as osmosis. Osmosis is the special case where water will diffuse through a semi-permeable membrane, which is what a cell membrane is. It's semi-permeable, meaning that some things are allowed to get in and other things are not. So to demonstrate this, we need a glass jar and we need a raw egg. And what we're going to do is we're going to fill the glass jar with vinegar. Vinegar is a weak acid, and I am not going to put the egg in now. However, I've done this already. If you put the egg into a jar of vinegar and let it stay overnight, you will find out that the vinegar, the weak acid, dissolves the eggshell. What you are left with is a shellless egg. Then I take the shellless egg and I put it into a jar of water. Because the concentration of water is greater on the outside than it is on the inside, water will then diffuse through the membrane of the egg, and the egg will begin to swell up. So here is a jar of water that I have put the egg into, and now I am going to pull the egg out, and we're going to measure how large the egg is. As you can see, there is no shell on this egg, but it is intact nonetheless. So I'm going to take a piece of rope and see how wide it is in diameter. It's this wide, which is approximately 21 centimeters in diameter. I did the same thing with an egg, but this time instead of putting it into a jar of water, I put it into a jar of corn syrup. Corn syrup has absolutely no water inside. Therefore, diffusion and osmosis will take place in the opposite direction. water will leave the egg and it will go into the corn syrup, resulting in a shrunken egg. So I'm pulling the egg out. You can see it is much, much smaller than the other egg. It has lost water. And measuring this, we get a diameter of, it's a little tricky because it's sticky. Okay, we get a diameter of 17 centimeters. So in water, it swelled up to 21. Without the water, it shrunk down to 17 centimeters. So this demonstrates the process of osmosis, that water will move through a semi-permeable membrane and go through an egg membrane, or in one direction or in the other direction, depending upon the medium it is in. Again, this is a very important biological process. Water goes in and out of cells all the time. When the concentration of water is greater in the egg, we say that the egg or the cell is hypertonic. When the concentration is greater on the outside, we say that the membrane, the cell, is hypotonic. And if it's exactly the same, which is what we're supposed to have, we call that isotonic. And that is it for today's episode. Bye all.